Good evening, all of you friends. Good evening, very good evening to every one of you. So, friends, in this particular class, we are going to complete the photosynthesis that we are doing the dark reactions. We are going to finish the entire dark reactions and we will see the questions also. Good evening, everyone. Very good evening, everyone, friends. Good evening. Please continue. Please continue. Okay. So friends, listen to me for the period of 10 minutes continuously, 10 to 15 minutes continuously. So immediately I'll solve all your doubts. So next one we called, we completely understood the light reactions. And friends, what is the ultimate goal of the photosynthesis? I hope all of you remember, friends. The ultimate goal of the photosynthesis is what, friends? Reduce the carbon into glucose. Everyone please continue. But we all read very clearly, if the plant want to reduce the carbon into glucose, what does the plant require, my friends? Undoubtedly, friends, plant require the energy actually. So that energy we have prepared already in the light reactions. Now energy is ready, my friends. Now what we have to read, what we have to do, good evening everyone. We have to just convert the carbon into glucose. So friends, to convert the carbon into glucose, we did not require any kind of the light. What we only require is what, friends? Energy. Always remember that point. So that's why we can call it as a dark reactions. My friends, dark reactions doesn't mean that they are occurring in the dark. Dark reactions means that we will be calling them as a light independent reaction. Or my friends, we will be calling it as the carbon fixation reaction. Friends, we can also call it as what, friends? Carbon assimilation reaction. You can call light independent also, you can call it carbon fixation also, friends. Even you can call it as the carbon assimilation. What do you mean by the word carbon assimilation? We are incorporating the carbon into the body to convert into glucose. So friends, all of you listen my statements very carefully, friends. Based on the dark reaction, what, friends? Based on the dark reaction or based on the carbon fixation, all the plants present on this planet Earth, quickly I'll recall, they are broadly categorized into three major types. All the plants present on this planet Earth, one we call a C3 plant, one we call C4 plants, one we call it as the CAM plants. All of you listen, I have told you in the last class also, friends. Listen to me carefully. 95 to 97.5% of the plants present on this planet Earth, they will be following the C3 plants only. They will follow the C3 pathway only. Friends, C4 pathway, the plants which are exclusively living at the high light intensities, which are living at the high light intensities, they will follow the C4 pathway. And my friends, CAM pathway, especially the plants which are living in the desert conditions, they will follow the CAM pathway. So friends, okay, now listen every one of you from this particular slide. Next 6 to 7 slide, whatever I am going to discuss about the C3 plants, I am requesting all of you pay utmost attention about every single slide. If the question comes, question is going to come from here only. Question won't go beyond that level. So I am encouraging everyone, pay attention towards that. By that you can easily understand the concept. I will explain in a utmost simple and elaborative way. Okay, let's focus. Dark reaction, pay attention for 15 minutes. Friends, you only try to understand, friends, in the light reactions what we have done. Good evening everyone. In the light reactions, friends, we have produced the ATP, and friends, in the light reactions we have also produced the NADPH. Now this particular ATP and NADPH which we produced in the light reaction, what the plant is going to do, good evening everyone, please continue. By using these two, plant is going to reduce the carbon into glucose. Very simple point it is. My friends, all of you please kindly keep it in your mind. When the plant is basically fixing the carbon into glucose, plant never fix the carbon directly into glucose. Instead of converting directly into glucose, first it can produce triose phosphates. Anyway, we are going to read it. Now friends, we all people read very clearly the light reactions where it has been taken place. The light reactions are occurred on the thylakoid membrane. We can also call it as the lamella. My friends, then where this particular dark reactions are taking place, friends, the dark reactions are exclusively taking place only in the chloroplast stroma. Why, you can ask me, sir, why the dark reactions only is taking place in the stroma? The reason behind that, every single enzyme, my friends, pay utmost attention. Every single enzyme required for the dark reaction, they are present in the stroma only. So that's the major reason you all people has to understand dark reactions are taking place in the stroma only. Now all of you remember friends, what is the unique enzyme that is involved in the dark reactions? We will be calling it as friends Rubisco. What is the full form of Rubisco? Ribose 1,5 bisphosphate carboxylase and oxygenase. Friends, why I am using a word carboxylase and oxygenase? The reason behind that, my friends, Rubisco fix the carbon also, friends. Rubisco fix the oxygen also. So it is having the ability to fix both. That's the major reason we will be calling it as the carboxylase also, friends. Even we will be calling it as the oxygenase also. Pay utmost attention. I'll make it extremely simple. You will understand and you can remember for a very long time. So, carbon dioxide assimilation occur in the 3 stages. Stage 1, stage 2, stage 3. What is stage 1? Listen to me 10 minutes continuously. There are 3 steps are there: fixation, reduction, followed by friends regeneration. All of you look into this. What is the ultimate goal? Once again remember, the ultimate goal is what? Reduction of the carbon into glucose. This is the ultimate goal. But how we are reducing? We are fixing the carbon into what, my friends? Into 3-phosphoglycerate. This is what exactly, friends, we will be calling it as the fixation. Now friends, this 3-phosphoglycerate can be converted into G3P. We will be calling it as the reduction. Ultimately, my friends, we will be calling it as the regeneration. I will explain everything. Friends, listen. For all of you, I will explain the cycle twice. What I am expecting for you is just your better understanding. I'll explain twice. Undoubtedly, you will understand it. Pay attention. As I already told you, friends, the ultimate goal is what, friends? Conversion of the carbon into glucose. This is the ultimate goal that we have already discussed. Now all of you listen. So we have to fix the carbon into glucose. If I want to fix the carbon into glucose, someone has to accept the carbon. Listen to me, friends, focus. Someone has to accept the carbon. Who is going to accept the carbon? The carbon can be accepted by RUBP. Don't forget, RUBP is called as what, my friends? A primary carbon acceptor. What do you call, my friends? Primary carbon acceptor. We called it as RUBP. Friends, everyone pay utmost attention. RUBP is how many carbon? 1, 2, 3, 4, 5 carbon. So 5 carbon RUBP accept 1 carbon. So 5 plus 1 will become how many carbons? 6 carbon. But here comes the point. In case of a Calvin cycle, the stable product and intermediate product is how many carbon? 3 carbon. Once again listen. The stable product or the intermediate product is the 3 carbon. All of you pay utmost attention, friends. Now this particular 6 carbon containing compound can be break down into what? This 6 carbon containing compound break down into 2 molecules of 3-PCA. This is what do you call carbon fixation. Now every student listen my words once again. Who is going to accept the carbon? The carbon can be accepted by the RUBP. RUBP is a 5 carbon, accept 1 carbon, total 6 carbon, giving rise to 2 intermediate molecules. Each one is the 3 carbon, we call 3-PCA. But let me tell you one interesting point here. What is that interesting point? Listen everyone. This is very unique statement. If I want to produce half glucose, I'm not talking about complete glucose. In order to produce a half glucose, the Kelvin cycle has to turn 3 times. Friends, never, if you forget this point, you can't understand the numerical part. In order to produce half glucose, my friends, cycle has to turn 3 times. So if one time 1 RUBP, then 3 times 3 RUBP. We already read, my friends, 1 RUBP is going to accept 1 carbon. Then, my friends, 3 RUBP is going to accept how many carbon? 3 carbon. 1 RUBP is how many carbon? 5 carbon. So total how many RUBPs are there? 3 RUBPs are there. So total how many carbon? 15 carbon. So each RUBP is accepting 1 carbon, 15 carbon plus 3 carbon, 18 carbon. My friends, now every one of you pay utmost attention. As we read that our stable product or intermediate product should be 3 carbon containing compound only. My friends, now we are having how many carbons in our hand? We are having 18 carbons. By using this particular 18 carbon, how many intermediate molecules I can produce? Undoubtedly, my friends, I can produce the 6 molecules of 3-PGA. Simple point it is. So 6 molecules of 3-PGA are going to be produced. Simple step. This is what do you call carbon fixation. Okay, now what is the second step? Everyone pay attention. Second step we call it as a reduction. What is happening in this particular step? There will be no change. Friends, here I told you 6 molecule of 3-PGA is present. Friends, each 3-PGA is how many carbon containing compound? Each 3-PGA is 3 carbon. 1, 2, 3. How many 3-PGA are there? 6 are there. Friends, during the process of reduction, everyone pay attention. 6 molecule of 3-PGA can be converted into 6 molecule of G3P. Friends, is there any change in the number of carbon? Each 3-PGA is how many carbon? 3 carbon. Each G3P is how many carbon? 3 carbon. Is there any change in the carbon number? Not at all. Now you can ask me, sir, what happened then? Just friends, 3-PGA just get reduced to the G3P. That's all. Friends, in order to reduce 1 molecule of 3-PGA, listen to me carefully. In order to reduce 1 molecule of G3P into, 3-PGA into 1 molecule of G3P, we require 1 molecule of ATP, 1 molecule of NADPH. But my friends, how many molecule of 3-PGA I am going to be converted into G3P, friends? I am converting 6 molecule of 3-PGA into 6 molecule of G3P. So my friends, I will use 6 molecule of ATP and 6 molecules of NADPH. Simple step. Now the toughest step. Every student has to understand this, and without confusion, you people has to focus. How many G3P are there finally, friends? 6 molecule of G3P are there. My friends, out of 6 molecule of G3P, how many G3P are left? How many G3P are involved in the production of glucose? Everyone understand, friends? Out of 6 molecule, only 1 molecule, just one molecule of G3P exit the cycle and it is going to produce a half molecule of glucose. Once 1 molecule of G3P comes out, my friends, how many molecule of G3P is actually left? You all know very well, 5 molecules of G3P is left. A very simple point. Friends, this particular 5 molecule of G3P, each G3P is how many carbon? 3 carbon. Very simple. So 5 molecules means how many carbon? 15 carbon. Now you only understand by using, listen my statements every one of you, by using 5 molecule of G3P ORR, by using 15 carbon, how many RUBP we can regenerate? Undoubtedly, my friends, we can regenerate the 3 molecules of RUBP, because each RUBP is how many carbon? 5 carbon. And we are having how many carbons in our hand? 15 carbons in our hand. So by using 15 carbon, I can easily produce the 3 molecules of RUBP by spending 3 molecules of ATP. This is the Calvin cycle. I am repeating. Everyone pay attention and every one of you has to solve my simple questions. If you answer my questions correctly, you can answer 2022 paper questions also just like that. Very easily. Now listen to me. Listen to me carefully. In order to produce half molecule of glucose, the Calvin cycle has to turn 3 times. So 3 molecules of RUBP, each RUBP is a 5 carbon, total 15 carbon, combined with 3 carbon, 15 plus 3, 18 carbon. 18 carbon catalyzed by the Rubisco, and it can be broken down into what? 3 carbon containing compound. How many 3 carbon containing compounds? We will get 6 molecules we will get, because we are having 18 carbon. This is the step we call carbon fixation. Now my friends, this particular 6 molecule of 3-PCA can be reduced to 6 molecule of G3P. How, my friends? By spending 6 molecules of ATP and spending 6 molecules of NADPH. Now friends, this part out of 6 molecule of G3P, friends, 5 molecules of G3P, or remember in this way, out of 6 molecule 1 G3P come out and produce the glucose. Then remaining 5 G3P, total how many carbon? 15 carbon, going to give rise to 3 molecules of RUBP by spending 3 molecules of ATP. Now every student has to answer my simple question without spending much time. My question is very clear. My question is, how many molecules of ATP, how many molecules of NADPH, how many molecules of carbon dioxide are required to produce 140 molecules of glucose? My question is very easy. How many molecule of ATP, how many molecule of NADPH, how many molecule of carbon dioxide is required to produce 140 molecules of glucose? My friends, I don't give them much time, just 1 minute, less than 1 minute. Everyone has to calculate faster, because we have to cover so much. Everyone, at least one fellow give me the answer correctly enough. Then I'll explain for all of you. Faster, friends. Just like that you can calculate. Anyone? Grishma, Amar, Beauty Kuwari, Sumaya, everyone, Priya, Sadguri, everyone has to answer. Jayashri, Deepika, Shahani, anyone? Come on, come on. Okay, that's okay, that's okay. I will be telling you answers. Just look into this. I did not ask any difficult question, right? Okay, all of you listen. How many molecules of carbon dioxide is required for half glucose? How many carbon dioxide required for half glucose? Half glucose, my friends, we require 3 carbon dioxide. Then for 1 glucose, how many carbon dioxide we require? We require 6 carbon dioxide. 6 into 140. Very simple, fantastic, very good, friends. And where most of the students will get confused is, when I ask you half molecule of glucose, 90% of the students will think that we are spending only 6 molecules of ATP. This is a wrong statement. We are not using 6 molecules for half glucose. We are using 6 plus 3. 9 molecules of ATP we are using. So for half glucose 9 ATP, then for 1 glucose 18 ATP. So 18 into 140. Then friends, half glucose, how many NADH we will use? 6 NADH we will use. Then for 1 glucose, how many NADH we will use? 12 NADH we will use. So 12 into 140, how much? You have to calculate. That's a simple calculation. You no need to get confused at all. Okay, 840 carbons. Now my friends, every one of you look into this. Everyone. Good. Some people given the correct answer, some people given the wrong answer. To summarize to all of you, just listen. 3 carbon dioxide combined with 3 RUBP to make 6 molecule of G3P. We studied very correctly. Out of 6 molecule of G3P produced, 1 molecule of G3P exit the cycle and goes towards making glucose. Now everyone pay attention, look into this. We were written very clearly here. Friends, 9 ATP are converted. Most of the students will think that 6 ATP. That is wrong statement, friends. 9 ATP are converted to 9 ADP, where we used 6 ATP we have used during the reduction step, friends, and 3 ATP we have used during the regeneration step. Then how many NADH we used, my friends? 6 molecule of NADH we used. So I am summarizing, friends, to make one molecule of glucose. If you remember for one molecule enough, you can calculate whatever they may ask. To produce one molecule of glucose, we require 6 molecule of carbon dioxide. Friends, we require 18 molecules of ATP, and friends, we also require 12 molecules of the NADPH. Once again, everyone, to produce one glucose, 6 carbon dioxide, 18 ATP, 12 NADPH, into how many number they may ask, you can easily multiply it. Simple. My friends, now I am giving you just 1 minute. Every one of you think and answer this particular question. It's a very good question, very logical question. I am expecting at least some people will give me the correct answer. Not much time, 30 seconds. Don't take too much time, friends. 30 seconds, that's all. What is the net synthesis of G3P and carboxylation of 3 molecules of RUBP in the Calvin cycle? Anyone? Not much time, not much time. 90% of the students will give the wrong answer for this question. I hope you people will think correctly and give me the right answer. Think it, friends, think it. You can think it easily and you can answer. 1. Saima said 1. Okay, what about others? Okay, Tiwari said 6. Okay, faster, friends, faster. Others, Beauty Kumari, Priya, Voya, Grishma, 6. Half glucose, Shafia, half. We did not ask the glucose, dear. Okay, anyone else? Sumaya? Yes, yes. Oya said 1. 6, Amar. Okay, my friends, all of you understand the answer is 1. Use your brain. Listen my question carefully. The question says that what is the, all of you. Okay, whomever made it wrong, listen my statements carefully. Listen, friends, look into this. The question says that what is the net synthesis of G3P on carboxylation of 3 molecules of RUBP in the Calvin cycle? My friends, every student has to remember when they ask you a question, what is the net synthesis? No problem, everyone look into this. What is the net synthesis? Every one of you has to understand. Net synthesis means how many molecules of G3P are directly involved in the production of glucose. Listen my question. How many molecules of G3P are involved in the production of glucose directly? You only look into the picture, my friends. We got 6 molecule of G3P. Out of 6 molecule of G3P, how many molecule of G3P is involved in the production of glucose? You can look into this. Only 1 molecule of G3P is involved in the production of the glucose. Very, very simple point. So the answer will be only 1. The question says that what is net synthesis? Net means which are directly involved. So answer will be 1. It's a simple question, logical question. Some people said 6. That is right. Because we produced 6 molecule of G3P, my friends, that is right. But out of 6, how many we used for the production of glucose? We used only 1. That is what do you call net synthesis. I hope everyone of you understand. Okay, now everyone of you answered this question which was asked in the February 2022 question paper. This is the first part B question from the photosynthesis or the plant physiology in the February 2022 paper. Everyone. Yes, Saima, what's the answer, dear? Deepika, Amar, Voya, everyone. Sumaya, Shafia, answer is D. This is the first question asked in the exam. Obviously the answer will be D. Simple 2 marker. What is, you can see, right, my friends, where we have used our energy. We have used energy during the process of reduction also, my friends, and most of the students will forget this. We have used the energy at the regeneration also. Very simple question. 2 marks are in our hand. Nothing difficult. Very, very simple. Okay. Now friends, pay attention, pay attention. Now this particular half molecule of glucose, what it is going to do? Friends, this particular half molecule of glucose, it is going to give rise to the sucrose also, my friends, and this particular half molecule of glucose, it is going to give rise to the starch also. Once again, this question also asked in the exam twice in the part B. What is the question they have asked in the exam? Which of the following are the locations involved in the production of sucrose, and which of the following location is involved in the production of the starch? Friends, please don't forget, glucose is what? Monomer, right? So glucose can give rise to the dimer also. Glucose can give rise to the polymer also. Now glucose is going to give rise to the sucrose. What you people has to remember for the exam point of view is location. Always, my friends, sucrose is synthesizing in the cytosol, my friends, where the glucose is going to be converted into the starch in the chloroplast stroma. I will be asking you the same questions in the tomorrow class also. Don't forget, while we are solving the PYQs and while we are solving the practice questions, you have to remember everything. Okay, that is the Calvin cycle. But my friends, from now onwards till next 10 minutes, listen my every single statement at most care. Why? Because many times we got the 4 marker question from here. Just listen to me carefully. Here, what is the principal enzyme involved, my friends? The important and the principal enzyme involved is the Rubisco. We can understand that. And friends, what is the uniqueness of the Rubisco? The uniqueness of the Rubisco is it can fix the carbon also, it can fix the oxygen also. So, but what is the ultimate goal of the plant body? Ultimate goal of the plant body is not to fix the carbon, not to fix the oxygen. Ultimate goal of the plant body is to fix only carbon, not the oxygen. But all of you listen, where certain conditions, what is happening, you know, Rubisco will fix the oxygen instead of carbon. So my friends, whenever Rubisco fix the oxygen instead of carbon, that reaction you called photorespiration. And every single student has to remember for the exam point of view. Most of the students will get confused that photorespiration can occur in the C4 plants also and CAM plants also. That is absolutely wrong statement. C4 plants and CAM plants never allow the photorespiration to occur. They will always try to avoid it by using their mechanisms we are going to read. Now, where this particular photorespiration is happening, only C3 plants. They're happening only in the C3 plants. Pay attention for 10 minutes. Don't interrupt. You will get a crystal clear concept. When this particular photorespiration is occurring, listen my statements carefully. It is occurring when the Rubisco fixes the oxygen instead of the carbon dioxide. What is the uniqueness of Rubisco? Having carboxylase activity also, oxygenase activity also. Now all of you can ask me a question. Sir, how can you say when the Rubisco fix the oxygen? How can you say when the Rubisco fix the carbon? I can give you a wonderful explanation for that. Rubisco is going to fix the oxygen only certain condition. What are that particular certain condition? Use the brain. It occurs only in the high light intensities. It occurs only in the high temperatures. It occurs in the areas of high light intensities. It occurs in the area of high temperature. Now you can ask me, sir, what is happening at the high temperature? Listen my friends, understand this beautiful line. At a high temperature, the solubility of oxygen in the cytosol and chloroplast stroma is less affected as compared to CO2. What do you mean by that? Friends, when the temperature is high, carbon dioxide will be completely solubilized. Then which is available? Available is only oxygen. Friends, oxygen is not completely soluble. Which is completely soluble? Carbon dioxide is completely soluble. Then which is left? Oxygen is left. then obviously Rubisco will fix what? Undoubtedly oxygen. Now once again, in a given 4-marker question, 2 times they have asked a same statement. You know what is the same statement they have asked? We are going to solve that. They asked that Rubisco having 2 separate active sites for the carbon dioxide and oxygen, which is absolutely wrong statement. It's not possible. You only try to understand. This is the enzyme Rubisco. Rubisco having which site? Only one active site. My friends, such a simple step it is. To this particular active site, carbon dioxide is competing, my friends. Oxygen is also competing, friends. So whichever associated with the active site, Rubisco only will fix that. You should not think that the Rubisco having 2 different active sites, by that it can fix the carbon dioxide and oxygen at a time. That's wrong statement. Whichever compete for the active site, whichever is more available, Rubisco only fix that. So in order to understand the photorespiration, right now whatever the 2 equations I am writing, if you understand that 2 equations, photorespiration is crystal clear to all of you. If you understand the 2 equations, whatever I am writing, the concept is clear. Listen. So first let's take, who is the carbon acceptor? Carbon acceptor is the RUBP. RUBP accepted what? Carbon. Now what is the enzyme here? The enzyme is the Rubisco. So here you know what the Rubisco has done? Rubisco fixed the carbon only. No problem. RUBP is 5 carbon. Rubisco is 1 carbon. Total 6 carbon. We already read. Then how many molecule of 3-PCA we are going to get? 2 molecule of 3-PCA. Then Calvin cycle will continued without fail. It will continue. But you listen, my friends. Now RUBP, same reaction. Now there is a no carbon, friends. The available is what? Only oxygen. Then same enzyme. What is the same enzyme? Rubisco only. Now RUBP is a 5 carbon. Is there any carbon available here? No carbon, only 5 carbon. Now you only answer my question, friends. By using 5 carbon, how many 3-PCA we will get? My friends, we will get only 1 molecule of 3-PCA. Then how many carbons are left, friends? 2 carbons are left. This 2 carbon containing compound, you know, my friends, what do you call it as? You have to remember this, friends. We call it as the phosphoglycolate. Listen to me, listen to me. We call it as phosphoglycolate. Now once again I am revising. When RUBP fix the carbon with the help of Rubisco, we got a 2 molecule of 3-PCA. Calvin cycle never continued, it will go on. But when the RUBP fix the oxygen in the presence of Rubisco, what is happening? We get only 1 molecule of 3-PCA. Now you only understand, my friends, if 1 molecule of 3-PCA is present, there is a no way the Calvin cycle will be continued. Only 1 3-PCA is left. As a result, what happened? Calvin cycle will stop. Now you can ask me, sir, what should I do in order to allow the Calvin cycle to continue? Here how many carbons are left? 2 carbon. What is that 2 carbon? Phosphoglycolate. Everyone listen to me. We have to convert this particular phosphoglycolate back to what, my friends? I have to convert the phosphoglycolate back to 3-PCA. Then only Calvin cycle will be continued. Otherwise, Calvin cycle is not going to be continued. And friends, here we produce a 2 carbon containing compound as a waste product. That's why photorespiration, also called as, my friends, C2 cycle. Simple point, na? Since the byproduct is a 2 carbon containing compound, then I will be calling it as the C2 cycle. So friends, C2 cycle is same, friends, and photorespiration is also same. Kindly do not confuse. All of you look into this. Oxygen and carbon dioxide compete for the same site on Rubisco. This is the same line they repeated in our exam more than thrice, not twice, thrice. Now you only understand, when Rubisco fixes the oxygen, we will get 1 molecule of 3-PCA and 1 molecule of 2-phosphoglycolate. That is why we can call it as a metabolytically useless product. And this entire photorespiration is only waste reaction. This should not happen actually. Okay, now you only try to understand. Now listen. what happened right now? All of you pay attention and understand whatever I'm writing. RUBP, taken the oxygen in the presence of Rubisco. I got one molecule of 3-PZA and I got one molecule of 2-phosphoglycolate I got. What should I do in order to allow the Calvin cycle to be continued? I want to convert this phosphoglycolate to the another 3-PZA molecule. Then I will get 2 molecules of 3-PZA. Then the Calvin cycle will be continued. Now I am going to prove how can we convert this particular 2-phosphoglycolate back to the 3-PZA. And you people no need to remember not even a single structure also. Once you download the PPT will be very clear. Don't worry. Just listen my words. First thing, you don't need to remember any structure. What you have to remember is schematic presentation. You have to remember every single step. Now listen. First, what are the various organelles involved? The organelle involved is the chloroplast, peroxisome, mitochondria. Friends, more than 6 times in a given 4-marker question they used to ask the organelles. You people has to remember the organelles in the order. Remembering order is utmost important, friends. Chloroplast, peroxisome, followed by the mitochondria. Listen to me, friends. I will explain in a simple way. Focus. RUBP fixed the oxygen. I got one molecule of 3-PZA. I got one molecule of 3-phosphoglycolate. Now how I am going to convert the 2-phosphoglycolate back to the 3-phosphoglycerate? Everyone, friends, just listen my words carefully. Now the 2-phosphoglycolate listen my words. The 2-phosphoglycolate upon losing the phosphate group, it is converted into glycolate. Now the glycolate from the chloroplast, it entered into the peroxisome. This is also asked question. Now in the peroxisome, glycolate converted into glyoxylate. Glyoxylate in the peroxisome, it is converted into glycine. Now the glycine from the peroxisome, it entered into the mitochondria. Now the glycine converted into serine. Glycine is an amino acid, serine is an amino acid. Now the serine from the mitochondria, it entered into the peroxisome. Now the serine converted into hydroxypyruvate. Hydroxypyruvate converted into glycerate. Glycerate from the peroxisome, it entered into the chloroplast. Glycerate, my friends, ultimately it is going to converted into 3-phosphoglycerate. Simple point it is. Very simple. I am summarizing for all of you. Pay utmost attention. What is the first step? 2-phosphoglycolate upon losing the phosphate group in the chloroplast converted into glycolate. Glycolate enter into the peroxisome. The question asked in our CSIR Part B exam. The first metabolite that entered into the peroxisome is the glycolic acid or glycolate. Absolutely true statement. Now glycolate converted into glyoxylate, glyoxylate converted into glycine, glycine from the peroxisome enter into the mitochondria. Everyone remember this particular reaction, my friends. Glycine is going to be converted into serine. Now everyone pay attention. When glycine is basically converted into serine, we gain energy here. It's a reduction reaction where we produce NADH. But what I said to you, I said it's a waste reaction because we have to spend energy. Listen my statement. I said to you, it's a waste reaction because we have to spend energy. But here, surprisingly, we gain the energy. Just listen that. Now glycine converted into serine. Now serine entered where? Into the peroxisome. Once again serine converted into hydroxypyruvate. Hydroxypyruvate converted into glycerate. My friends, when the hydroxypyruvate is going to be converted into glycerate, this is what kind of reaction? Oxidation. So here we have to spend energy. So whatever we gained, whatever the energy we gain, we spent here. Chart balanced. Now glycerate from where? From the peroxisome it enter into the chloroplast. Now the glycerate is going to be converted into the 3-phosphoglycerate. My friends, ultimately see here we produce 3-phosphoglycerate. Now you only understand. If the glycerate wants to be converted into 3-phosphoglycerate, obviously what should I add? My friends, I should add the ATP. So here we are spending energy unnecessarily. We have already discussed in the light reaction, plant struggled a lot to produce the energy. Now whatever the energy we synthesized over there, we are spending unnecessarily here. That's why we will be calling this particular photorespiration or the C2 cycle as the expensive side reaction. This is how, friends, the C3 plants can show the C2 cycle or the photorespiration. Anyway, undoubtedly, we converted the glycolate back to the 3-phosphoglycerate and we have skipped this particular photorespiration. Now, you have to get one wonderful doubt. What is the doubt you have to get? Sir, does this particular this is the real good doubt. Does the photorespiration occur very oftenly or very rarely in the plant body? I can tell all of you, in the C3 plant, photorespiration occur very oftenly, because you only understand, friends, when the temperature is high. Even we are also one of the reason, because of the human activity, what is happening, you only tell me, the temperatures get increased. When the temperatures get increased, what is happening, friends? The carbon dioxide will get completely solubilized. Then which is available? Oxygen is available. Then the plant fixes the oxygen. Then obviously it leads to the photorespiration. So you should not think that the photorespiration is a rare phenomena. Photorespiration is the more often phenomena in the C3 plants. Okay, friends, everyone say yes or no. Is that clear, everyone, till C3 cycle and C2 cycle? If you say yes, we will go ahead. If you ask me doubt, I will explain. Quick. We have to finish 2 more pathways. Faster, friends, faster. These pathways are easy. Okay, I hope it's clear for every one of you, friends. That's great. You can ask me, friends. You can ask me if you have anyone having any kind of a doubt. Now, friends, let's talk about the C4 pathway and CAM pathway. And friends, you have to remember these 2 plants, C4 plants as well as the CAM pathway plants, they never ever involved in the process of photorespiration, never ever involved. So they will always develop certain kinds of mechanisms, friends, where they are going to avoid this particular photorespiration in a very easy manner. Let's talk about one by one pathway. Very easily I'll explain, and very depth we will read. You will be very clear. And first, let's talk about the C4 pathway. So friends, C4 pathway can be shown by what kind of plants? I want every student has to remember, my friends, more than 18,000 types of angiosperms. More than 18,000 type of angiosperms are basically follow this particular C4 pathway, especially monocots. But my friends, there is an exception. 99.99% monocots will follow the C4 pathway. But friends, at 0.1001%, even dicots will also follow this particular C4 pathway. The best example is Amaranthus. Amaranthus is the dicot which can follow this particular C4 pathway. Please kindly do not forget this particular point. Now, C4 pathway are plants. Just listen to me. 10 minutes I'll explain in a elaborative way. C4 pathway are plants, where the plants that are growing in the hot and dry climate. Whenever you heard the name of a C4 pathway, one of the most common point everyone has to remember for the exam point of view is that C4 plants are those type of plants which are involved in the Cranch anatomy. This statement also asked more than 3 times. C4 pathway contains Cranch anatomy. Absolutely true point. There is a no doubt. I will tell you what is this particular Kranz anatomy, how exactly it look like. And one more unique statement is what, friends? Very, very unique statement. In case of a C3 plants, the principal enzyme is the rubisco. But my friends, in case of a C4 plants, the major principal enzyme is what is that, friends? We will be calling it as the pepper carboxylase enzyme. Use pepper carboxylase to fix what, friends? Atmospheric carbon dioxide. Never ever forget that. And it will fix the carbon dioxide into how many carbon containing compound? 4 carbon. This is interesting point. Why we have given the name C3 pathway, tell me? Because the stable or intermediate product is the 3 carbon. That's why we call C3 pathway. Here the stable or intermediate product is 4 carbon. That's why we can call it C4 pathway. What is the best example for that? We call it that 4 carbon containing compound is the oxaloacetic acid. Simple point. What are the best example? Sugarcane, maize, sorghum, millet. These are all the 4 standard example. I request every student at least remember another 4 plants apart from these standard 4 plants. Friends, now I want every one of you look into this beautiful diagram. This is the Kranz anatomy. Try to understand, friends. See this, how beautiful this. This is the upper epidermis of the leaf and this is the lower epidermis of the leaf. Please listen my statements crystal clearly. If you look into the Kranz anatomy, it contains 2 type of cells. One we called it as the mesophyll cell, another one we called it as the bundle sheath cell. My friends, what are the major differences between the mesophyll cell and bundle sheath cell? Everyone listen. Mesophyll cells are those type of cells, friends, which are always present very close to the leaf surface. You only see here, this is the surface, right? Mesophyll cells are always present to the leaf surface. You have to listen this. And the unique statement, it is present very closer to the leaf surface and having great amount of the intercellular spaces. Great amount of the intercellular spaces. And one more important point is what? If you look into the bundle sheath cell, all of you pay attention. They are present in a very deep. If you don't understand this statement, you can't get the concept. Bundle sheath cells are present very deeper and they are not having any intercellular spaces. You can see the diagram very clearly. Once again, look into this. Mesophyll cells are having they are associated with the leaf surface and they are having large intercellular spaces. Whereas, friends, bundle sheath cells do not have any intercellular spaces. They are associated very tightly. Now you can ask me, sir, in the C4 pathway, which kind of cells are involved? Do we require mesophyll or bundle sheath? The point here, we require both mesophyll also, we require both bundle sheath also. Both the cells are extremely important. So you can ask me, sir, you are saying that 2 cells, both mesophyll and bundle sheath is involved. Then the question is here, how these 2 are connecting with each other? That is the question. My friends, mesophyll and bundle sheath cells are connected with each other by the plasmodesmata. Every one of you remember, both the cells are required. Desmata, with the help of plasmodesmata, they can be connected. So friends, before understanding the pathway, just listen to me carefully. Every one of you pay utmost attention. This is the mesophyll cell. This is the bundle sheath cell. Friends, I just said to all of you, this is important. For 5 minutes, pay attention. Mesophyll cells are very closer to the leaf surface. I have shown you in the diagram very clearly. The interesting point every student has to remember is that whatever the cell that are very closer to the surface, they do not have rubisco. Simple point. If there is a no rubisco, someone has to fix the carbon. We should not stop the carbon fixation. So who is going to fix the carbon? My friends, the another unique enzyme, phosphoenol pyruvate carboxylase, is the enzyme which is involved in the fixing of the carbon. Now here comes a beautiful and interesting fact about the PEP carboxylase enzyme is that it cannot fix the oxygen. It cannot fix the oxygen. It is going to fix only the carbon. That's a great advantage. Now, whatever you will understand in the diagram, just listen to me. Whatever the carbon fixed by the PEP carboxylase, it will send it to the bundle sheath cell. Friends, bundle sheath cell, what is present? In the bundle sheath cell, Rubisco is present. Now you will get immediately doubt. Sir, you are sending carbon to the bundle sheath cell, there is no problem. And there is a Rubisco also, that's an advantage. But Rubisco can fix the oxygen also, right? Then how can you confirm me that the Rubisco only fix the carbon? The greatest point here is that bundle sheath cell oxygen never diffuse, because bundle sheath cell is presented very deeper to the deeper layers. Oxygen having a poor access, it cannot diffuse, hence there will be a no process of oxygen fixation. Now I will summarize it. Listen to me very carefully, everyone. Who is the carbon acceptor? My friends, everyone pay attention. The carbon acceptor is the PEP, phosphoenol pyruvate. Don't confuse, my friends. In case of a C3 plant, who is the carbon acceptor? In the C3 plant, carbon acceptor is the RUBP, which is the 5 carbon containing compound. But my friends, in case of a C4 plants, the carbon acceptor is the PEP. PEP is how many carbon? Just 3 carbon. PEP combined with this particular carbon. So PEP 3 carbon plus 1 carbon, how many carbon we will get? 4 carbon. What is that particular 4 carbon? We will be calling it as we will be calling it as the oxaloacetic acid. My friends, we all people know very well oxaloacetic acid is a 4 carbon, hence the name of the pathway also C4 pathway. But the point every student has to remember, friends, this particular oxaloacetic acid cannot enter into the bundle sheath. Cannot enter into the bundle sheath. It do not have active site actually. My friends, for that reason, oxaloacetic acid can be converted into either malic acid or the aspartic acid. My friends, malic acid also 4 carbon. There is no doubt about it. Everyone pay attention. Now the malic acid entering where? The malic acid is basically entering into this particular bundle sheath cell. It entered here. Malic acid is how many carbon, my friends? 4 carbon. Now friends, malic acid can be treated by an important enzyme. All of you pay attention. We called it as the malic acid decarboxylase enzyme. My friends, we know that decarboxylase enzyme function is what? It is going to break down the malic acid 4 carbon into 3 carbon plus 1 carbon. Now here comes the unique one. This 1 carbon taken by the Rubisco, because Rubisco is present in the bundle sheath, right? Now friends, Rubisco continued the Calvin cycle. Calvin cycle is common. It has to occur in every plant. Do not forget that. Now how many carbons are left? 3 carbons are left. What I can call that particular 3 carbon? I can call that particular 3 carbon as the pyruvate. Now that particular 3 carbon containing pyruvate entered where? It entered into the mesophyll cell. What is that? Pyruvate. Now my friends, what is happening? This particular pyruvate, with the help of ATP, it is converting into what, friends? Phosphoenol pyruvate. So wherever the cycle started, it ends there only. Now you only listen. Is there any fixation of the oxygen here? No, not at all. Is there any occurrence of the photorespiration here? No, not at all. So that's why we can say C4 pathway plant, by using Cron's anatomy, they can easily avoid the photorespiration. Once again, every one of you pay attention. I'll quickly summarize. Phosphoenol pyruvate is a 3 carbon containing compound. Combined with 1 carbon, total become 4 carbon, giving rise to oxaloacetic acid. Oxaloacetic acid converted into malic acid. Malic acid from the mesophyll enter into the bundle sheath cell. Malic acid can be treated by an enzyme called as malic acid decarboxylase enzyme. So malic acid decarboxylase enzyme will break down into 1 carbon plus 3 carbon. What is that? No, I am not understanding. Someone is asking. Then this particular 3 carbon pyruvate can enter into the mesophyll, and pyruvate is converted into phosphoenol pyruvate. Oh, I got it. So this is what we call C4 pathway. I understand. Okay, now everyone say yes or no. Is that clear, everyone? C4 pathway? You can ask me, friends. Everyone can ask me. C4 pathway is clear. Just say yes or no. Okay, I hope it's clear for everyone, right? Perfect, perfect, perfect. I hope it's extremely clear for all of you. Now, my friends, then the last and final pathway that we have to study is the CAM pathway. My friends, undoubtedly, in case of a CAM pathway, also we have to avoid the photorespiration. There is no doubt about it. In the CAM pathway, there is a two problems. What are the two problems? First problem, we have to avoid the photorespiration. Oh yeah, I will explain once again, dear. Please wait. We have to avoid the photorespiration, and my friends, in the CAM pathway, another goal is what, friends? Another goal is to minimize the transpiration. I will explain, Jayashree. Minimize the transpiration and avoid the photorespiration. There are two goals are there for the CAM plants. All of you pay attention, friends. Pay attention and listen to me very, very carefully. Now, what kind of plants are going to follow the CAM pathway? The plants which are belonging to the Crassulaceae family. All these plants follow the CAM pathway. Best example, all the desert plants like a cactus, pineapple, bromeliads, agave, succulents. Remember many. And what is the unique point you have to remember? We have to avoid the transpiration. We have to avoid the photorespiration also. Friends, listen my interesting statement here. If you observe the temperature, I will repeat, just listen to me. Please listen, please listen. Have patience. I'll explain. CAM pathway. All of you listen. In case of a CAM pathway, friends, we have to avoid the photorespiration and we have to avoid the transpiration also. And what is the specialty of CAM plants? They are growing in the desert. Now, whenever they are growing in the desert, everyone pay attention. Whenever they are growing in the desert, if you observe the temperatures in the desert, during the daytime, the temperature is very, 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 very high. But my friends, when it comes to the night time, the temperature is dramatically fall off. It will be extremely cool. In the night time is extremely cool. In the daytime, it is extremely hot. Now you only understand, use the brain. If a CAM plant open their stomata during the daytime, what is going to happen? If it opens during the daytime, obviously what, friends? Entire water loss will occur. So that is the reason. What is the uniqueness of the CAM plants? They will open the stomata only during the night time. They never open the stomata during the daytime. Whatever the stomata that is opening the night time, everyone has to remember the name. We will be calling it as the sunken stomata. Sunken, S-U-N-K-E-N, sunken stomata. Just everyone listen, I will explain twice. So this is the condition where the stomata is opened, that is the night time. This is the condition where the stomata closed during the daytime. Very simple, friends, listen. Now what is the function of stomata? We all know very well the function of the stomata is the gaseous exchange. So when the stomata is opened, gases are coming inside. So carbon dioxide enter inside. All of you same function. No difference. No difference, just listen to me. Carbon enter inside. Who is going to accept the carbon in case of a C3 plants? In the C3 plant, carbon is accepted by RUBP. Like this also we can get the question. In C4 plants, carbon accepted by PEP. My friends, even in the CAM plant also, carbon is getting accepted by the PEP only, friends. Now the phosphoenol pyruvate accept one carbon, 4 carbon, same enzyme, no doubt. PEP carboxylase. So PEP carboxylase convert this into oxaloacetic acid. Everyone answer my question. In case of C4 pathway, what happened, my friends? Oxaloacetic acid convert into malic acid. Same way here, oxaloacetate also convert into malate. My friends, listen. Now in case of C4 plants, what happened? Malate entered into which cell? Bundle sheath. But friends, where are the bundle sheath? No bundle sheath in the CAM plants. Then the malic acid can be stored in the vacuole. Where the malic acid is stored, friends? In the vacuole during the night time. Whomever did not understand, once again listen to me. When the stomata opened during the night time, carbon enters inside. Phosphoenol pyruvate accepts the carbon. With the help of PEP carboxylase enzyme, it is converted into oxaloacetate. Oxaloacetate converted into malate. Malate or malic acid, they can store it in the vacuole during the night time. Don't confuse. Malic acid is the organic compound. Malic, it is an acid. One is acid or organic compound. Anything is same. Now listen. During the daytime, what happened? Stomata is closed. If the stomata is closed during the daytime, carbon never enters inside. But the point is very clear. Plant has to survive. Plant has to survive. Without food, they can't survive. Now see what they are doing. They have stored the malic acid during the night time. Whatever the malic acid that they stored during the night time, they will bring that particular malic acid out. And once again, they will be treated with an enzyme called as malic dehydrogenase enzyme. What is happening? Malate is how many carbon? 4 carbon. So it breaks down into 3 carbon plus 1 carbon. Very simple. Now that 1 carbon can undergo the Calvin cycle. That's so common. Then what about the 3 carbon? 3 carbon is the pyruvate. Pyruvate is going to be converted into starch. Simple. Now you only try to understand, my friends, where the photorespiration occur? Not at all. We did not fix the oxygen. We fixed carbon only. Moreover, what has happened? Moreover, there will be a no transpiration. There will be a no water loss. Whomever did not understand the CAM pathway, I'll summarize in 1 minute. Focus. CAM pathway having 2 goals: avoid the photorespiration, minimize the transpiration. So for that reason, they open the stomata during the night time. Carbon enter inside. Phosphoenol pyruvate accept the carbon, convert into oxaloacetate. Oxaloacetate convert into malate. Malate can stored in the malic acid. Now all of you listen. This particular malic acid during the daytime, it will come out. Right, then malate once again break down into 3 carbon pyruvate and 1 carbon carbon dioxide. Pyruvate convert into starch. 1 carbon undergo the Calvin cycle. Simple CAM pathway. Sahani, that's really very good question, dear. Enzymes are going to damage at higher temperature. That's absolutely true statement. But you also know, no dear, 97.5% of all the enzymes are stable at 37.5 degrees centigrade. But the enzymes which are present in the plants, which are living at the desert condition, they can withstand up to 70 to 80 degrees centigrade very easily. Hence, there is no problem for that particular enzymes. Now, whomever did not understand the C4 pathway, I am repeating for all of you, and I will explain one by one. And someone has asked a doubt, and Beauty Kumari, you look into your doubt first. I will come one by one. First, phosphoenol pyruvate combined with 1 carbon, 3 plus 1, 4 carbon. 4 carbon converted into oxaloacetic acid. Oxaloacetic acid converted into malic acid. Malic acid entered here. Malic acid is how many carbon? 4 carbon. Once the malic acid convert is a 4 carbon, it is treated by an enzyme called as malic acid decarboxylase enzyme. Malic acid decarboxylase enzyme break down malic acid into 3 carbon plus 1 carbon. That 1 carbon can be used by the Rubisco and Calvin cycle will be continued. Then how many carbons are left? 3 carbons are left. Now the 3 carbon, what do we call it as? Pyruvate. That pyruvate from the bundle sheath cell, it enter into the mesophyll. Now all of you listen. Now pyruvate is going to be converted into phosphoenol pyruvate. Now if the pyruvate wants to convert into phosphoenol pyruvate, we have to add the phosphate group. Then only pyruvate convert into phosphoenol pyruvate, right? So how can we add the phosphate group? Someone has to donate the phosphate group. Who is going to donate the phosphate group? ATP. So simple. Friends, is that clear everyone? I hope all of you clear now. If you look into the C3 plants, there is only mesophyll cell. If you look into the bundle sheath cell, both mesophyll, if you look into the C4, both mesophyll and bundle sheath. If you look into the CAM plants, only mesophyll. But you know, right, stomata opened during night time, stomata closed during daytime. You know, my friends, you call it this as the temporal separation. Please kindly do not forget this, temporal separation. Yes, I think Tarun, your doubt is very clear. Now itself in this slide, it is only contain mesophyll. Okay, that's about photosynthesis. I hope everyone is very clear with the photosynthesis without having any confusion. Now you can ask any kind of a doubt in the entire photosynthesis. I hope it's clear, friends. That's all for now. We will meet in the tomorrow class. Okay, and friends, tomorrow we don't have the early morning 5:45 class. 5:45 classes I will start from the Wednesday. Okay, so first I have to finish two extra classes are there. I have to finish that. From Wednesday onwards we will start the 5:45 a.m. classes. Okay. Thank you all of you. Thank you so much. That's all for now. I'm telling you once again, I will remember every one name and I will take utmost care of everyone when it comes to the when we are going forward. I will help individually by providing the study materials also. So I'm requesting all of you friends, please be regular. I don't expect anything. Just be regular and have patience. Okay friends, thank you all. Thank you so much. Good night. Bye bye. Okay, Sadhguri, I will tell that in the tomorrow starting class, dear. How many ATP? I will tell you in the tomorrow starting class.